Okay, report. Good. Okay. Yes. So yeah, I'll give this introduction to the project. Yeah, our project is called COFOLD for.
Ebbe?
Yes.
We only see a black screen now.
Oh, okay, that's not good.
We had the image before, but now it's just black.
Okay, maybe that's weird. So you don't see anything changing now? No? Okay, so maybe yeah, I'll just do a reshare. I'll stop the share and then I'll do a reshare. Maybe sharing the PowerPoint directly. Yeah, that's sometimes better.
Yeah, that looks good.
Okay, wonderful. Yes, good. But yeah, so the main title is co-transcriptional folding for RNA medicine and synthetic biology, and I'll try to explain what this actually means. Yeah, first of all, here's a little overview of our team. Yeah, so we are these 4 groups involved. Yeah, and yeah, Victoria has gotten Sophia Thompson started as a PhD. She's here today, and then I think a postdoc position will be posted at some point. It is posted, yes, and a person will pop up eventually. Then Ariane and Adam here in yeah in Leiden at Leiden University, and then yeah Christian and Johan started his PhD just recently, working on bioinformatics. And then, yeah, here in my group, we also just gotten started actually. Or we have been working because Kalinka has been working on Cryo yeah, since the start as a research assistant. And then Emil started something like yesterday. And then we had actually some problems getting a PhD student through in the PhD school, but yeah, that will eventually kind of succeed. We hope. So yes, good. So yeah, you might be asking yourself, what is RNA and co-transcriptional folding? So I'll just yeah, kind of give a little introduction to that. So it's basically yeah this process of yeah how an RNA polymerase kind of reads and transcribes an RNA from the DNA template, and that is this process of how the RNA folds up fast during the transcription process. There's some timing things here that actually yeah extension is rather slow and the folding is much faster. So that basically means that the RNA folds yeah before it's completely finished. So there yeah, and then of course when it's formed and finished, it can form larger structures. Yeah, I kind of put a few review articles down here that might be useful to get a little intro to yeah both what is the importance of co-transcriptional structure formation. There's also a really nice recent review on thoughts and how to think and talk about RNA structure, just to get you into the lingo of and understanding of yeah how complex RNA structure actually can be, because of course yeah, so some people just have the idea that it's just a wavy line in the textbooks, but it's actually like a really complicated molecule that can fold up into very intricate shapes. And then there's also a review from a group in the US, the Lux lab, that actually works a lot on co-transcriptional folding, both experimentally and computer simulation wise. And they're mainly working on biological systems and trying to understand those. But he has written a recent review that really gives a nice overview of what this process is and why it's important to study. So, and this is exactly also yeah, what is the motivation to study this particular problem? Why is co-transcriptional folding important? So I tried to put a few perspectives here. First is a biophysics perspective that we really want to understand in more detail how biomolecules fold during synthesis, and it's this problem of yeah, one needs to understand the folding kinetics, and it's a non-equilibrium system. So normally when people have been thinking about folding of molecules, it's always like yeah, just one strand compacting on itself as a equilibrium thermodynamic system. And here, what biology does is completely different. It basically folds like gradually and so on. And yeah, so and somehow we need a better understanding of this. Then there's a molecular biology perspective for trying to understand biology. So yeah, basically, yeah, understanding these folding processes is also a way to more deeply understand yeah, how the complex molecular machines in nature are made, like the ribosome. It's a really complicated gradual process to form these complex molecules. And then I guess my own perspective is a bit on the engineering side, because yeah, we have been developing like RNA nanostructures and so on, and by doing that, like designing from scratch, you somehow also need to learn all of these rules about like how RNA and proteins come together, and then also engineering and technology perspective is also somehow to then exploit that knowledge to some extent, yeah, to basically design molecules with specific functions. And here then also comes in the application perspective that if we can succeed in designing these structures, yeah, more kind of with better control and with higher quality, then we also have the possibility to improve, like, yeah, production of RNA medicine, mRNA vaccines, and so on, and also to synthesize some of these devices. And because they're co-transcriptional folded, they can also be like integrated in a cell and be expressed inside a cell. So that also means that we can, yeah, basically, yeah, redesign properties of cells, and that's a field called synthetic biology. Okay, so the project that we kind of are working on here, yeah, has the main aim of studying the co-transcriptional folding process, and the main idea is actually to try to use designer molecules instead of natural biomolecules like the ribosome, because studying the ribosome would be like a very complicated place to start. So here, our basic hypothesis is somehow, okay, if we can design small like toy systems, then we can much easily kind of yeah, dissect the different like mechanisms and properties. And then here, the idea, yeah, also because it's this interdisciplinary setup, we basically try to combine expertise from bioinformatics, from single molecule FRET studies, from cryo-EM and cryo-electron tomography. And here, the aims are then distributed on the different groups. So in one is Christian and Johan going to work on studying prediction and design algorithms for co-transcriptional folding. Yeah, and then yeah, possibly there's some kinetic parameters needed for predicting or simulating this, and this is then kind of investigated by Victoria's group and Sophia to determine folding kinetics by single molecule FRET. So single molecule FRET has this like really high time resolution, and by studying like simple systems, maybe we can yeah, dissect some of the folding kinetic parameters. Then it's our group who wants to study time resolved folding by cryo-EM. Here, the main idea is kind of to freeze the folding reaction at different time points, and then yeah, study the folding intermediates. And then yeah, we're also going to kind of use other techniques on top of that to support these studies. And then there's Arian and Adam studying in vivo folding by cryo-electron tomography. And here, tomography is the ideal method to actually see the molecule in its natural environment. So not all in just in the test tube, and this can add a lot of interesting insight, I think. So yes. Good. So then back to presenting our group. So I just did a little slide here to show our lab here. And yeah, we have a like general research topics on RNA nanotechnology and synthetic biology. And then we use yeah, different biophysical characterization methods. Currently, we have two projects going on in the lab. One is the COFOLD project here, where we are. There's one guy here who's actually not here today, and he's not directly a part of the project, but his name is Cody Geary, and as you'll see in some of the coming slides, he has been one of the inventors of the RNA design or origami method. So he might kind of help us out with design along the way. Then there's Emil just started, and the PC and Kalinka, and we have two bachelor students starting soon who will work on related topics. Then we have another kind of Novo Nordisk funded project as well that's more into synthetic biology, where we try to apply these RNA structures in yeast and try to use it to organize enzymes to kind of facilitate enzymatic pathways. So yes, good. So, but it somehow all builds on this, or the work in our group very much builds on this method called RNA origami that we developed initially at Caltech and interacted a bit with Ariane over there to actually study some of the first our first tries on designing these structures by EM. And at that time, it just looked like spaghetti, I think. So we were not that successful initially, but then finally, we got it to work. And it's basically this software that allows you to build a RNA structure on the computer, to maybe build a 3D model, so you can kind of iteratively build up the structure you want, and then some sequence design algorithms that from this 3D structure can give you a sequence that you need to order. When you order the sequence, you can transcribe it in the test tube, just adding a polymerase and some nucleotides, and then the structure is formed, and you can use different methods to characterize it. And we initially kind of published it back in 2014, but then actually then during the years and while using it in the lab, we have developed some new improved software that makes it even better. So the main design problem somehow here that relates to co-transcriptional folding is that when you build up such a structure, then you have to somehow choose the strand path of how the RNA, one single RNA, is going to like route through this maze of RNA structure. And what we found out, is that it really makes a big. It's very important, like which path you choose, because it can easily get entangled. And but then you can also choose a path that gets less entangled. And with some of these, both software and also these rules, we've been able to kind of increase the scale of these RNA structures quite significantly, up to about 2,000 something structures. But also, as you see here with the white numbers, the yield kind of goes down as we increase the structures here towards the size of a small ribosome unit. But basically because now we are starting to get into trouble with the folding path. And we've done some earlier studies also where we kind of have then like tested different paths and different like designs with our algorithm, and then can see that it affects the yield quite a lot. Depending on how you do this path. So it's actually not like super kind of big changes here. So actually this path produces like maybe 30%. It's also, we chose on purpose a difficult path initially, but then if we choose another path that has a lot of topological issues, then we see a decrease in the folding yield, and also a lot of misfolded structures in the EM images. And then if we do it worse with the sequence design, it all goes goes bad. So and then when we have designed some of our scaffolds, then we can start using them as organizing scaffolds for proteins. I won't get too much into that. Then lately we've been then working during the last 3 years, I would say, working on extending the design pipeline by a characterization pipeline using now cryo-EM instead of atomic force microscopy, as you saw before. The problem here with atomic force microscopy, as you saw up here, is that the structures kind of fall flat on the surface, and and they're somehow maybe look more ideal than what they really are in solution. So cryo-EM single particle averaging method is really good to be able to see the molecules as they are in solution. And I'll just show you a few examples here. So we had some success in getting this pipeline running. So actually putting a lot of different RNA structures through the pipeline, both origamis and actually also some Emil brought an interesting sample from Cambridge, an RNA polymerase ribozyme and other interesting structures. I'll just show you one example here. This is one of the tiles that we got the highest resolution on, where we now can go in and now the motifs here are colored. So there's some kissing loops here in purple, some junctions in blue, and some tetra loops in yellow. And we initially see that our this design was supposed to be completely flat, but we see it has a certain curvature to it. One thing we can see is that if we kind of decrease the threshold level, we can start almost seeing the bases. And if we mask a certain part of the structure, we can get high local resolution down to 3.4 angstrom, and then look at these motifs that are important for the structure. So here's an example of a kissing loop motif, and we can kind of see exactly how it's composed. We could also figure out that actually our initial design principle was a bit wrong. This kissing loop takes up less space than we originally thought, and so on. So in this way, we can really learn something from the cryo-EM structures. Also, the junctions we figured out had a very specific geometry. And then the nice thing is that we could then use that to basically correct our initial designs. So it's actually this one here out on the very right, where you can see that we go from that the tile has a certain twist of minus 28 degrees, and now we can try to correct it to something like 15 degrees by just changing a few parameters. And also the structure now looks more flat and more perfect. Good. A last story I just want to tell is we've then been looking at some bundles of this origami, and when we would have had a spatial design, we observed a fun thing in the samples that actually in the same sample this bundle would have two different conformations. So after 5 hours, it would actually be 50% of one and 50% of the other. And when you kind of look at a cut through or a side view, you can see that it kind of initially is quite open with this bridging helix kind of going across. But then suddenly it kind of changes to a more compact conformation. So we think there's some kind of maturation process going on, and we could kind of find out by overlaying the structures and morphing them, as it's called. Let's see, here it goes. That then we could actually see what's going on, that somehow, like this kissing loop bridge breaks, and then when it breaks, it suddenly can become more compact. So this is also just an example of a folding kind of process. It's not co-transcriptional folding, but it's still a consequence of co-transcriptional folding, because this trap that we call it a kinetic trap is formed during the folding process, and then it's so stable that it actually lasts. You can see here for about 6 hours before it then changes conformation. So this is an interesting property that RNA can kind of make very stable intermediates, and here we're doing some small angle X-ray scattering, where we can see that there's an initial kind of compaction process going on, and then there's this more kind of dramatic change, and then actually also after this kissing loop has broken, there's also a further compaction, and here it's something to do with how the helices like to pack against each other, and that's a more slow process. Okay, and then I just want to mention that as another study that we then did on this bundle together with a group. I think you can maybe see them up here. It's a guy called Gary Wren at the Molecular Foundry at Berkeley, and they are also doing cryo electron tomography, but here they specialized on doing it on single particles. So here they were able to analyze our sample exactly at that time when the transition happens. After 5 hours, they were able to then capture or freeze the sample at that time. And then they could see all the intermediate states, both this initial compaction, also some very rare states where the kind of structure is completely like flat actually. And then also here between 44% and 68% is actually where this breakage of the kissing loop happens. And then they could also see a further compaction afterwards. So of course this is a special technique where they somehow process the single molecule tomograms and in different ways to get the 3D structure. But I think it's quite fascinating here that you can actually get or see the single molecule with something like helix resolution, where you really can see where each helix is. So I think this method could prove important to study some of these kind of folding processes. Okay. So I don't know how much time I used, but I'll just tell shortly that we have the postdoc project that's kind of supervised by me and Ariane as a co-supervisor here, and the aim of Emil's project here is to study the non-equilibrium self-assembly process of this folding process on design structures, and here the idea is both to do something called shaped SIG. I think I have a little overview of what this is. This is basically a way where you can probe your RNA chemically, and you can then so this chemical kind of mainly modifies single-stranded regions, and then if you afterwards sequence this, you can kind of see where these chemical modifications are, and then you can get the approximate secondary structure of your molecule. And then if you do that at several different like time points or extensions of the RNA, you can get these maps where you can, like here, you have a short transcript on top, and then it extends, getting longer and longer and longer. And then as it gets longer, you can then see where are the yellow ones are, I think, the single-stranded regions. So you can see sometimes there's an initial region that then disappears, and so on, and you can in this way get an idea of the folding process over time. Yes. And then the other thing was to then try to do cryo-EM alongside that, freeze the samples at certain different time points, and then do initially single particle averaging analysis of these, and see if we can see some of these changes, and maybe also using this new cryo-ET method to study the whole population at different time points. But where the main goal is somehow to get experimental data on these folding processes. Yes, there's a little bit about how we plan to possibly do this time-resolved cryo-EM or tomography, where we both will try to sync the polymerases so that they all kind of are activated in sync. So you can, by leaving out one nucleotide, you can kind of have them sitting on the promoter ready to go. And then when you add the last nucleotide, they all like start simultaneously. And then if it's a big RNA, or if we decrease the amount of nucleotides, we can also kind of make the process go rather slow, so that we maybe on a second time scale can be able to stop the process during the production of the RNA. So and then there's the PhD project that we are still looking for a candidate for. So here the idea was to we actually thought that it would be like something that could attract PhD students, because we use this nanorobot word here. So the idea is to design RNA nanorobots that can sense, compute, and act in response to their environment. And the idea is somehow to use some of these co-transcriptional folding traps or products to help do some of these kind of mechanisms. So just to explain you how this could work, because we actually already have an example of this produced in the lab. Nestor did during his PhD an RNA origami that can be activated by two input keys, and then has a functional element that's initially kind of bent out of shape so that it doesn't work. But then when you release it by these two inputs, it turns on fluorescence. And he actually has been showing that you can, here it's just an opening and a closing of the device, but he also shows that you can do this multiple times, and also that it works a bit like an AND gate, that you need both of these keys to activate the fluorescence completely. So it's these kind of things that we would like to achieve, making RNA devices basically that can be activated in a very specific way, and this could be something that could be used for medicine. So you activate only your your molecular function under in a very certain cell, or it could also be devices that are expressed inside cells and then activate gene expression under very certain circumstances. So it's an example of application. But here this is with Victoria as a co-supervisor. So here the idea was also to combine cryo-EM and fluorescence spectroscopy and so on, FRET systems and so on, to try to develop some of these fun devices. Good. So that was actually all. I hope we recorded it. Okay, I think we did. Yes. Okay. Good. Yes. So
we have to do it again.
When so it's just when the future PhD arrives, then we can show it. Okay. Yeah. So please, if there's any questions or discussions, we can maybe it actually fitted quite nicely with half an hour. Thank you. So I don't know. So of course, I think of course one of the questions, is how to attack this problem. It's quite complicated, of course, to study this kinetic process of folding, because basically some of so I can maybe mention some time scales. So I think like an RNA, or one of the fast RNA polymerases, goes at about 150 nucleotides per second. So that's already pretty fast to be able to like stop the process with these techniques that we were talking about. But then of course, if you make not just an RNA of 150, but make one of the 2,000, 3,000 nucleotide long structures, also maybe make a rolling circle thing where it's actually continuously like producing a long ribbon of the RNA, then maybe you have a better chance of capturing some of these the process while it's going. Yes, Victoria.
The reaction should be limited by the availability of the reactants, right?
Exactly.
So
yes.
we can just speed.
Yes. Yep. And concentration of nucleotides in the that works quite well. There's also different polymerases. Like the one I mentioned here was the fastest known. So that the polymerase is working like in E. coli and in mammalian cells. They go maybe at 70 nucleotides or 20 nucleotides per second and so on. So that's also
the numbers that you quote, the numbers that are found out. The polymerase will work better if it's in a good environment with some proteins to help it work, or is it in vitro?
So I think some of these so it's like often old literature where they've been testing these enzymes in the test tube, but I think also they have very precise estimates from in vivo production of RNA. That's also quite easy to kind of get those numbers. But then the more interesting thing is actually that the polymerase the polymerization is rather slow, but the folding of secondary structures in RNA is on a microsecond time scale. So they basically form like immediately. So each new nucleotide has a long time to kind of find its or forms its thermodynamic stable state very fast. And yeah.
So that should make the time feel like it doesn't matter that we slow it down, or it doesn't enhance the code transcriptional folding too much if it's really fast anyway.
Yeah,
But that could be a problem.
say,
like if you slow it down, then perhaps it will happen more than it would naturally.
yeah. So I'm a bit in doubt about the kinetics. How it's going to affect the speed of the polymerization. It's going to affect the folding itself. So I can just say that what we have been looking at until now is actually not so much the speed of things. Or it is indirectly, but it's mainly that like certain structures can fold and make a topological trap so that new sequence that's made cannot somehow enter and form the base pairs because of the structure already formed. So yes, but that basically happens because we're also using these kissing loops, which are tertiary interactions. So initially we have the secondary structure forming, but then these strong kissing loops that we're using are also forming fast during the transcription. So they can basically lock the structure into a suboptimal fold for later folding. So it's these things that we are kind of playing with.
I just wanted to say that I having read a little about it now, I think it looks like it's two different like time regimes. One of them is the secondary structure formation, and then it's the tertiary structure formation.
Yeah.
Secondary might be less than seconds,
Yes.
and tertiary might be half an hour.
No. But tertiary can also mean a kissing loop, and a kissing loop forms also less than seconds.
Okay, so but then what I mean is that we could probably get into a structure, and then that can relax over a long time. So we are looking at two different regimes. We're looking at either slow relaxing or really fast relaxing. And then what they do in the loop step is actually not that they slow down the polymerase. They just have a transcript that stops at kind of all the positions along the way.
Yes.
So that you have a polymerase that's like goes
a certain length. Yeah.
And then they shape on all the different lengths of the sequences.
So that's not so much like they don't capture the process while happening, but they just produce different lengths of transcripts and then see how does that length fold basically.
And I think it's going to be difficult to find the fast folding.
Yes. I think also that there's some early studies out there where especially a researcher called Sarah Woodson has kind of been trying to get at some of these very fast processes, but by using some very like some kind of synchrotron to kind of get the right like free radicals to somehow be able to probe the structure or the flexible regions at a very fast time scale and so on. So I also think that actually being able to do of course the dream would be to like on the microsecond time scale be able to somehow see the process in action, but that's I guess almost impossible, and would require a very like advanced setup. So I think also maybe we should somehow define what what we want to target. If it's the long, of course, it's more easy to target some of the longer time scales. But on at the same time, it could be possible maybe to study the speed of this kissing loop, which is a tertiary interaction. How fast does that actually happen? And so if we take some very toy examples, we might be able to kind of get time parameters out on certain kind of interactions that happen on the second time scale.
just one thing that comes in mind. So one way to look at the fast secondary interaction would be to have just some hairpin forming, but then with a longer loop. And then you would see that at some point it starts being really slow, but in the beginning you can measure it's together already when it's formed, and then that's kind of a way to see how fast can it actually be.
There was also the project that you were looking at, Victoria, with some of the RNA motifs, like these fluorescent aptamers that have a certain tertiary structure with the quadruplex and so on, and then also somehow also just study how fast do those type of structures mature after being produced, and how long does it take to kind of find the right confirmation to do its job. Or, so and the other thing is Victoria's group is experts in the quadruplexes and so on. So it could maybe that would also be an approach to study some of the more tertiary folding mechanisms.
I had a question.
Can you hear in the other?
The kissing loop that you're showing,
Yes.
do you know what is the reality
We have kind of a
finding or
ballpark
interaction?
range. I think normally when we use it in our algorithm, we just calculate the delta G of the actual base pairs forming, but actually it's much stronger than that, because it's a tertiary interaction where certain base pairs stack and so on. I think I asked Cody recently, but he was just giving the standard calculation, but I think it should be found in the literature somewhere where people have been, or anyway, getting maybe better estimate.
A whole bunch of calculations on how to calculate the MT of the kissing loop, but it's very difficult.
Okay.
It's about the local concentration.
Yeah, as well. Yeah. But
So I imagine that theoretically it's already known, but now it's a kissing loop, it's origami design.
yeah, it's kind of
And I can imagine that the bases around can help stabilize and destabilize these interactions.
yes.
So I don't know if this is something we should start thinking about, or it's beyond the scope, or I don't know.
Yes.
That's where I was.
Yeah.
Like, I don't know if you guys are starting thinking about that, or
But it's definitely like when it's intramolecular, then somehow it's a cooperative process between many of these interactions, that they actually, if one forms, then it helps the other form and so on. But I think these things are very interesting to study. Not just understanding the kissing group interaction in solution, but when they are part of a larger molecule, how does the local environment affect the speed of that they find each other and so on. So So I know from some of these like people have been studying DNA origami, how it assembles and so on, and tried. It's very interesting for them to like see the cooperative effects of these many interactions working together. So it's something that's very interesting to study. Yes. Yeah. So Ariane, maybe what do you think about this cryo electron tomography on single molecules? So I can say that so first of all, the main paper that we've been doing with the origami cryo-EM and the trap, we just submitted the revision to Nature Nanotechnology yesterday, and now we hope finally that it gets through. But I should say that for some of the tomography on the single molecules, we had a lot of problems with some reviewer who was very skeptical. But we think that we now have kind of made him happy by kind of recording these images at low electron kind of dose, so that there should not be any electron damage. So that was his main complaint. but then of course also I think it's somehow not completely standard to kind of get the 3D structure from a single molecule without sub-tomogram averaging and so on. But when I've been looking into the algorithms that they use and so on, they it seems like they've been verifying all the steps with this missing wedge and so on, that they've been testing that the way that they solve for that kind of doesn't distort the some ideal molecules that they've been or some model molecules they've been trying it on and so on. But
so I'm not very familiar with the method he used, so I would have to look into this. This is not something we have done in our lab, and I think it's really not a standard thing to do in the field. So I'm not sure how much general how generally accepted it is. And I just remember when you mentioned like the electron doses that he used initially.
Yes,
they were insane. I mean, we are usually like quite high in our cellular tomography, but that was like even like much higher than what we would throw at a cell. And we know it does affect the ultrastructure quite a bit at those doses. So like, so what you mentioned, that was super high. I wouldn't trust anything as well, but like, so I don't know what his
so the new doses are like 68 and 120 electrons per square angstrom. So I think that was kind of more in the
it's more reasonable. I think if you want to push for a higher resolution, it's probably still on the high side. Like if you like you probably break every single bond at, I don't know, like already like at much lower doses, but that's at least more reasonable in what people typically use. 120 is like something typically for tomography, and I think 68 is makes more sense. But yes, I do not know those softwares that he used. It's not something we have done yet. We could look into this, but it's not something we've used before. Maybe a question, what the advantage of that is versus just standard single particle and proper classification. I'm not sure.
It's a good question, but it just I think you are somehow losing some of because an RNA structures have a certain they're quite dynamic and flexible. So it's not like a protein that maybe just has one like rigid structure or like a core fold. So I think somehow for RNA, definitely seems from our studies until now that we always have been not getting the highest resolution, because you're basically throwing a lot of molecules away to get that one conformation that's most representative, and then you can get down to 4 angstrom resolution. But then we can also do something called 3D variability analysis, where you can then by some principal component analysis kind of take a lot of more of the data into account, maybe going from 10% of the data to 70% of the data, and now you can start seeing the helices kind of move.
But I'm curious. Like, this is more, I think, for the single particle experts. But I think the field definitely is going more into like dynamics of, like also very dynamic proteins and like these, like very wiggly things in a way from these very stable rocks. And so I wonder what the best way forward is in the future for these RNA structures, especially with, the new detectors and the new microscopes that can collect much more data. So I'm not sure what the best way forward is actually with this. If you're better with single particle and classification and just collecting lots of data, or if this tomography, single particle tomography is something that will be useful. But I think why not try both?
I somehow think looking at those single molecule tomography data that they show much more that what you can get out of averaging, because you see rare states and maybe transitions that you would never capture in or like by averaging maybe.
But I think if you look at the labs that do push these, you know, the methodology in these dynamic systems, I think you get much more in those data sets as well.
Yeah, that's true.
Yeah.
And another thing that we so at the conference in Copenhagen was that that there seems also to be a lot going on with combining cryo-EM and fluorescent microscopy.
Yeah. Yeah, absolutely. And we do have so at the moment we do have like a cryo stage for, so you can, you know, investigate the same grid with fluorescence and then put it in a microscope.
Mm.
So we do have this set up here.
Yeah.
So we could totally try that.
What type of microscope do you have for like microscopy?
So we have a Leica Thunder with a cryo stage.
Mhm.
And we will also get it's called a MIMA system, which will be like a FIPMIL instrument with a fluorescent inside. So if you want to, you know, like look at thick cells and thin those and do correlative light microscopy at the same time, we will be able to do that as well.
So is that a confocal microscope or is it wide field or what?
So it's not a confocal. That would be nice.
Yeah.
Yeah, but I just see some because some of the origami structures that we're making, we can kind of fluorescently label and so on. So it's there seems to be some connection there between making some
maybe something that, you know, that might be interesting a bit down the road is that, I mean, people have done also a super resolution in cryo and then combined with tomography. So we've done that in Grant Jensen's lab like already a few years ago.
Mhm.
So, you know, that might be interesting down the road as well.
Mhm. Great. Yes. So so maybe another thing that we could, apart from the more science part, is also maybe talk a bit about how we can get discuss some individual projects so for example Ariane and I could meet with Adam and discuss his project and the other constellations. Could be nice to kind of have some discussion about plans and so on. Yeah.
Speaking of that, Ebbe, since you're Adam's co-supervisor, he has to do a presentation for the for the group.
Okay.
And it would be great if you could be there online. If that fits your calendar, and I think the one is scheduled on December 20th.
Okay. Yeah. Great. I'll try.
That would be great.
Yeah, this is December 20th. Yes.
Yeah.
I would love to.
Awesome.
Great.
Yes, I will send you the details and then we'll log you in. But that would be great. I meant to ask you like many times and I keep forgetting. December is so far away.
Yes. Cool.
But maybe, for example, Ebbe, maybe we can have like a cryo meeting for, you know, the projects that involve that, like with the, you know, also with the postdoc and Adam, and like maybe we can have that
Together,
because maybe I have some ideas, maybe what you could try with your time resolved ideas. So
yes. That would be really nice to get that running. That's great. Good.
Maybe I can also say that the stuff I want to do with shape, I'm thinking about doing kind of a library of doing many sequences so that you can get something for you guys that where you can look at sequence variation and how it goes. And of course there'll be the time resolved aspect of it as well. I don't know if that's something that makes sense or otherwise I should.
It depends very much on the actual setup. Like we talked about like last program for the like the project meeting. Lucas also mentioned it, that what I was also in doubt of, like if you only make point mutations, that it's not going to affect the thing enough that I could get any data from it, which actually, for my purposes, be more helpful to get bigger random mutations, and they maybe not be at all exhaustive, but it usually doesn't have to be.
You can easily get many mutations, but you can't have a big library. That's
No.
So I think just the general thing here is that we should think about synergies between the projects. So what we are talking about here is that Johan and Christian are thinking about some machine learning methods, and there they really need like huge amount of data, and maybe maybe we can produce them for them by some of these probing, folding by sequencing, and so on, and then also we could maybe normally machine learning people kind of just take the data that's available in databases, but maybe here we could kind of make it for the purpose.
So how about, so can I suggest something? Like we have another project here that's like a big project with 6 PIs and lots of students and postdocs. And what we did is that the students and postdocs actually had a Slack or WhatsApp group. And so it will, I think, help the students among each other, like maybe brainstorm and chat. And it might be nice that they have the way communicating without the PIs observing. So maybe I would suggest maybe that would be
freedom of speech. That's good. Yeah, that's a good idea, Ariane.
I can make a WeChat app if you want.
Okay.
So I think that would be nice, right? I think it works well. I talked to my student and she really likes that, and they can exchange ideas and speak freely, and I think some good ideas came from that.
Perfect. What about synergy? Yeah, so I think there's some synergy between barium and like resonance microscopy that we could try to kind of investigate if there's ways to obtain kind of data on that can somehow support each other, but then and then we also already are working on synergies with the resonant aptamers and so on. So I think
but I think so from experience with a student group, there were lots of synergies that came from that just by chatting and bouncing ideas around. I think maybe that would be a good idea to see if that will help.
There was people locally at the same place, right? Because I think it's probably a little more difficult when we're far apart, but of course we should do it.
So right for generation.
Yeah, in our case it's not local. Like we're like also 4 different universities that are quite far away, and so the students just share this WhatsApp group, and this is how they started also, like forming little collaborations and really bringing the team together in a way that I think we couldn't do if it's just the PIs constantly talking. I think it is helpful, so we could try. We could see.
Great. Is there anything else that people have on their mind or questions about like basic things that we should kind of great, but else I think let's close the meeting, and thank you so much for joining in, and let's one thing I can say is that I was planning to kind of maybe send out a little email once in a while, maybe once a month, so maybe just to kind of but both of us, if there's some updates about our COFOLD project, but maybe also to suggest some papers on the topic and so on, so people can kind of get a common knowledge base on what it's all about. So
yeah, I think that would be great, especially so the 3 papers you had in your presentation, maybe you could send those around.
I'll send those around. Another thing I can just say that I was thinking about was that I was thinking about maybe trying to arrange some kind of online seminar about RNA folding, or it could maybe be a bit broader topic, but the idea was to invite this guy Julius Lux to give a talk where we could get some kind of experts in the field. And I think the experts are kind of spread out in the world. There's also some people in Japan who works on code transcription and folding and so on. But maybe an online forum for this could be interesting. And I think also it's wrong if we just ask them to give talks for our little group. So I think it's better to find some kind of more general idea where these guys or women also kind of gains from it, so that we have a little bit of focus on this topic. I'll also try to kind of kickstart this. I think we were. I think actually I did a laptop page. I think it's pretty empty. Good. So we have one. And then we will ensure that you get access to it. So that's a nice place just to, so everything is like on a wiki and get stored in an orderly way, so it's easy to find later, and not, you don't have to dig into your big inbox. 1,000 million mails. Good. Okay. Thank you very much.
Thank you, everyone.
Thank you.
Have a nice day. See you.